bodies found in neutrophils. A. Remnants of rough ER, indicating toxic granulation. B. Peroxidase positive cytoplasmic inclusions. C. Nuclear fragments from megaloblastic change. D. Blue-gray RNA remnants of rough endoplasmic reticulum. The correct answer is D. Dowell bodies are pale blue-gray cytoplasmic inclusions in neutrophils consisting of remnants of rough endoplasmic reticulum, RNA. They indicate bacterial infections and toxic states. A sputum culture shows a mucoid colony that produces a blue-green pigment with fruity odor. The organism is oxidase positive and non-lactose fermenter. Most likely organism is A. Klebsiella pneumoniae, B. Pseudomonas aeruginosa, C. Burkholderia cepacia, D. Acinetobacter baumannii. The correct answer is B. Pseudomonas aeruginosa produces pyocyanin, blue-green pigment, fruity/grape-like odor, is oxidase positive, and a non-lactose fermenter, all classic characteristics. In Jaffe's reaction for creatinine estimation, which compound is used as a chromogen? A. Diacetyl monoxime, B. Alkaline sodium picrate, C. Ninhydrin reagent, D. Diazobenzene sulfonic acid. The correct answer is B. Jaffe's reaction uses alkaline sodium picrate, picric acid in alkaline conditions, which reacts with creatinine to form an orange-red complex that is measured spectrophotometrically. The Weil-Felix test is based on cross-reactivity between rickettsial antigens and which organism? A. Proteus mirabilis OX19, OX2, OXK strains. B. Brucella abortus standard tube test. C. Salmonella typhi HNO antigens. D. Leptospira interrogans somatic antigens. The correct answer is A. The Weil-Felix reaction exploits antigenic cross-reactivity between Rickettsia species and certain strains of Proteus, OX19, OX2, OXK, which share polysaccharide antigens. Periodic acid, SHIF, PA, stain is negative for which of the following structures? A. Glycogen in hepatocytes. B. Fungal cell walls, Cryptococcus. C. Amyloid deposits in tissue. D. Basement membranes in glomeruli. The correct answer is C. PA stain detects polysaccharides, glycogen, fungal walls, basement membrane. Amyloid is a misfolded protein, beta-pleated sheet, and does not contain polysaccharides. It is PA-negative, positive with Congo red. A patient typed as group A Rh positive receives a cross-match. The minor cross-match tests donor serum against A. Patient's red cells, B. Donor's red cells, C. Patient's serum, D. Saline control only. The correct answer is A. In a minor cross-match, the donor's serum/plasma is tested against the patient's red cells to detect antibodies in donor blood that may react with patient RBCs. The Philadelphia chromosome, T9, 22, results in which of the following fusion oncoproteins? A. Pml fusion protein, B. BCR-ABL tyrosine kinase, C. AML1 ETO fusion protein, D. NPM-ALK kinase. The correct answer is B. The Philadelphia chromosome T9, 22, creates the BCR-ABL fusion gene encoding a constitutively active tyrosine kinase, which is the hallmark of CML and targeted by imatinib, Gleevec. Coagulase negative staphylococcus that is novabiocin resistant and the commonest cause of UTI in young sexually active women is A. S. epidermidis, B. S. lugdunensis, C. S. saprophyticus, D. S. hemolyticus. The correct answer is C. Staphylococcus saprophyticus is novabiocin resistant, unlike S. epidermidis, which is sensitive, and is the second most common cause of UTI in young sexually active women after E. coli. The reference method for measuring total serum protein is A. Bradford method using Comessi blue, B. Lowry method with phenol reagent, C. Biuret method measuring Cu superscript 2 chelation, D. Kagel-Dahl method measuring total nitrogen. The correct answer is D. The Kagel-Dahl method is the reference/gold standard for total protein as it measures total nitrogen content directly. The Biuret method is the routine clinical method. A stool examination shows cysts with 4 nuclei, chromatoid bars with rounded ends, and a glycogen vacuole. This is characteristic of A. Entamoeba histolytica, B. Entamoeba coli, C. Giardia lamblia, D. Balantidium coli. The correct answer is A. Entamoeba histolytica. Mature cysts have 4 nuclei with chromatoid bars with rounded, cigar-shaped, ends. E. coli cysts have 8 nuclei with splinter-like chromatoid bars. In hemoglobin electrophoresis at alkaline pH, pH 8.6, which hemoglobin migrates furthest toward the anode? A. Hemoglobin A, B. Hemoglobin S, C. Hemoglobin C, D. Hemoglobin H, beta 4 tetramers. The correct answer is D. At alkaline pH 8.6, HbH, beta 4 tetramers, migrates fastest toward the anode due to its high negative charge. Migration order, HAFSC, anode to cathode. Which media is selective and differential for isolation of Salmonella and Shigella from feces? A. McConkey agar, B. XLD, xylose lysine deoxycholate, agar, C. CLED agar, D. Thayer-Martin agar. The correct answer is B. XLD agar is selective, deoxycholate inhibits coliforms, and differential, xylose fermentation, lysine decarboxylation, H2S production. Salmonella shows red colonies with black centers, Shigella shows red colonies. In ELISA, the enzyme substrate system that produces a yellow product red at 450 nanometers is A. Alkaline phosphatase with PNPP substrate, B. HRP, horseradish peroxidase, with TMB substrate, C. Beta-galactosidase with ONPG substrate, D. urease with urea substrate. The correct answer is B. HRP, horseradish peroxidase, with TMB, 3,3,5,5 tetramethylbenzidine, substrate produces a blue color that turns yellow upon stopping with H2SO4, read at 450 nanometers. The most common ELISA system. The osmolar gap is calculated as, measured osmolality calculated osmolality. A gap 10 mosm/kg suggests A. Hypernatremia with pure water deficit. B. Presence of unmeasured osmoles, methanol, ethanol, mannitol. C. Diabetic ketoacidosis without alcohol. D. Pseudohyponatremia from hyperlipidemia. The correct answer is B. An osmolar gap 10 mosm/kg indicates unmeasured osmoles in the serum. Classically toxic alcohols, methanol, ethylene glycol, isopropanol, ethanol, or mannitol. The confirmatory test for paroxysmal nocturnal hemoglobinuria, PNH, in modern diagnostics is A. Ham's acidified serum test, acidified hemolysis. B. Sucrose hemolysis, sugar water, test. C. Flow cytometry for CD55/CD59 deficiency on RBCs. D. Direct antiglobulin test, DAT, with polyspecific AHG. The correct answer is C. Flow cytometry detecting CD55, DAF, and CD59, MIRL, deficiency on RBCs and granulocytes is the modern gold standard for PNH. Ham's and sucrose tests are older, less sensitive methods. Quiz complete. You scored 0 out of 15. Flexed biceps don't give up. Revise and retry.